Okay, so the last topic then for this semester, for this course, is metabolism. And you're going to find this in the first two sections of chapter 18. Now, in chapter 18, if you go beyond section 2, you will get swallowed up in the minutia of biology and biochemistry as they go into very great detail to explain each of the processes that we're going to describe very generically here today. And that's one of the reasons why we aren't covering them. But if that is something you're interested in, or if you remember it just from high school biology, oh, hey, we mentioned, I've heard that term before. What is that? The latter sections of this chapter will cover that, but we're not going to here. So primarily, we're going to be talking about metabolism, and we're going to be talking about how your body breaks down and digests the three different kinds of macromolecules: carbohydrates, fats, and proteins, and how it converts them into energy that your cells use to, well, live. And that's where we're going to stop. So in section 1 here, that's primarily what we're talking about: metabolism and ATP. ATP, adenosine triphosphate, is the energy molecule that your cells use. And ATP is the storage mechanism by which that molecule, those cells, we store energy as ATP, we break down ATP as we do certain kinds of biological processes, and then we resynthesize ATP from its components when we ingest and digest food. So it's a very continuous cycle of events that are taking place inside of our body. We have a molecule that has a high amount of potential energy. That potential energy gets put into use to run chemical processes. The remaining products, which have lower potential energy, get recharged when metabolism, when your body uses food. Now, metabolism itself is a very complicated series of chemical reactions. And we call it just under this generic category of metabolism, but includes any chemical reaction that is going to provide energy and the substances that are needed for your body to grow. And growth does not simply just mean growth like how a child grows. It can mean any kind of cell generation, regeneration process. So even as you get older and you stop growing physically taller, or going through any of those other kinds of developmental processes that you might have gone through as a teenager, your body is still growing in the sense that you are still creating new cells, you are still regenerating different parts of your body, and that will continue the rest of your life. Those kinds of reactions can break down into two categories. The catabolic reactions, these are reactions that break down large complex molecules that will provide energy, as well as smaller molecules, the byproducts of that complex molecule breaking down. So this is the kind of stuff that we talk about when we are seeing carbohydrates broken down by your body and absorbed into your digestive system to give you energy. Proteins being broken down, fats being broken down. Those are catabolic reactions. Anabolic reactions, on the other hand, take that ATP that was generated through the use of the byproducts of those catabolic reactions, and we use that ATP to help us build larger molecules in the process. So those are the two things that are happening. In terms of catabolic catabolism, there are 3 stages. The first stage in catabolic reactions is the digestion stage. So again, this is primarily you eat something, that something goes into your stomach, you begin to break it down inside of your body. The first stage is the digestion stage and the hydrolysis stage. The acid in your stomach starts to break down the complex biomolecules into smaller pieces and send off those smaller molecules into the bloodstream. The second stage is called degradation. In the degradation stage, we're going to see that those smaller molecules formed by that digestion process, formed by the hydrolysis of those bigger molecules into smaller molecules. So maybe we're talking about sucrose being broken down into the glucose and fructose. Well, in the degradation phase, that glucose and fructose gets broken down into even smaller molecules, 2 to 3 carbon molecules as opposed to 5 and 6 carbon. And then the third stage is an oxidative stage. And if you've taken biology at any kind of level other than freshman biology in high school, you have heard some of these terms before. You've heard of the citric acid cycle. You've heard of electron transport. Now, if the only thing you have in your background is high school biology, then ATP is probably the only thing here that you recognize, because that's kind of where they stop the process when we talk about photosynthesis. But that third process, that oxidation process, that's taking those 2 and 3 carbon molecules and breaking them down even farther. Oxidizing them, using the electrons that are coming from that oxidation process to do the finest, smallest kinds of functions that happen in your cells. That also happen to be some of the most important things that happen in your cells as well. Now, in all 3 stages, what we see is basically a degradation continuum. We start with the big macromolecules, the stuff that we ingest. Again, thinking back to kind of our conversations with our proteins, our fats, and our carbohydrates. We know that carbohydrates are big molecules. Even sucrose. Sucrose is a 12 carbon double ring structure. Starches are even bigger. And we know that a lot of the foods we eat are starchy. So you're talking about many of those glucose rings stacked around each other. We know proteins. Proteins can have hundreds of amino acids. Each one of those amino acids has 3 carbons in it, or at least 2. And fats, we know fats, triglycerides, we've got a 3 carbon backbone and a whole bunch of carbon tails hanging off of those. So we've got big molecules that we're starting with. So that's what we start with. That's what we eat. as we go through the 3 stages of catabolism, we see that the big molecules get a little bit smaller in the first stage, the smaller molecules get a little bit smaller in the second, and the smallest molecules get even smaller in the third. Every single phase ends up breaking down what was there before into smaller pieces. Now, if we are looking at this flow chart, and this flow chart is on page 620 of your textbook, our discussion is basically going to go into some detail in stages 1 and 2. We're going to leave stage 3 for the people who are really heavily studying this stuff. And that's not us. Because yes, you get into acetyl-CoA and you get to the citric acid cycle and you get into these other processes. And again, unless you're studying biology, like, for real, you probably don't need to know all of these things that are happening. And again, if you've seen this stuff, if these terms look familiar to you, well, there's a reason why. That's the reason why. If you've taken an advanced biology course, that's where you would have seen this stuff. But we're going to focus on kind of the top of the flow chart here. What happens in the digestion processes? What happens in the degradation processes that lead us to this oxidation stage? And then we're going to stop. Now, your book goes into some more details about kind of where each of these processes are occurring. And we're going to talk about them a little bit. We've already mentioned some of these terms. We talked last chapter about the nucleus and how the nucleus is the place where the genetic material is stored, and how the messenger RNA and the transfer RNA all come out here to the ribosomes to build proteins. That's stuff we talk about. A lot of these things involving energy actually happen in the parts of the cell called the mitochondria. You'll hear, I just saw someone mouth it. If you haven't heard it before, it is the powerhouse of the cell. Love that term. Powerhouse. But the reason it's called the powerhouse of the cell is because this is where the majority of these energy interactions are taking place. So just as kind of an overview, maybe a little bit of review, just again, going back to some of the things you've seen before. What are some of the major components here? Well, again, we've already talked about the nucleus, and we've talked about ribosomes being that's where proteins are made. The cytoplasm, that is the stuff inside the cell, kind of the aqueous liquid part of the cell. It is water-based. Stuff that exists in the cytoplasm has to at least be somewhat water soluble, or else it will not travel through the cytoplasm. The cytosol is the fluid part that actually will contain many of the enzymes that are used. The mitochondria are where we find the structures that allow us to create ATP as one of the byproducts of this digestion and series of metabolism steps. And it's the ATP that is ultimately going to do a lot of the lifting as far as when a reaction needs energy, the ATP can provide the energy that it needs. And then the cell membrane is what holds it all in. Cell membrane is a barrier, semi-permeable barrier. Helps keep the cytoplasm in, helps keep the external stuff around the cell out. But things are able to pass through that cell membrane selectively. And when we talked about osmotic pressure a couple of weeks ago, that was one of the things that we talked about, because water is one of those things that is able to pass through the cell membrane. And so solution dynamics can be a really important part of biochemistry and what happens in your cells. In your body, energy is stored up in what is called adenosine triphosphate, ATP. Now, if you're looking at this and saying, that looks kind of familiar, well, adenosine would be this nucleic acid backbone here. We've got the ribose sugar, we've got the adenine nucleic acid base group. What makes it different is that instead of this part being then hooked on to maybe one phosphorus to create DNA, we're hooking it to 3 phosphate groups. And so that's where the triphosphate comes in. We've got 1 phosphorus group, 2 phosphorus groups, 3 phosphorus groups. Now, there are variants of ATP known as ADP or AMP, and all those are just based upon the number of phosphates that are attached. So AMP, we're talking about just 1 phosphate group instead of the 3. ADP, we're talking about 2 phosphate groups instead of the 3. And the one that we're going to see the most often is the ADP. So ATP and ADP are very closely related to each other because it is in that conversion of ATP to ADP that energy gets released to allow the cells to maintain functionality or build molecules or drive the creation of different substances. Every time our cells need to do pretty much anything, whether that is perform muscular tasks or move stuff across a cellular membrane, have your body send nerve impulses from your brain to other parts of your body. Basically, again, anytime your body does anything, the energy to do that comes from ATP. And in particular, it comes from the hydrolysis of ATP. So if you remember what hydrolysis is the breaking of a bond through the insertion of a water molecule. So if we think about ATP, and for lack of drawing skill and simplicity, I am going to draw the adenosine here as a diamond and the phosphate groups as circles. Okay. In a hydrolysis reaction, what we would get is the insertion of a water molecule to break that bond here and give us ADP. And now a free phosphate molecule. Now, ordinarily, when we break a bond, that actually requires energy. At least it requires heat energy. Most of the time when we break bonds, heat energy is required to do so.
Don't we need energy to break bonds? And then don't we get energy to break bonds?
Well, so if we are looking at things from a heat standpoint, usually when a bond is broken, that is going to require energy because we have to break the associations of those two atoms to each other. And obviously that would require energy to do. And when we bring things together, the mutual attraction between them usually results in excess energy being let go. So usually, again, thinking about things more from a heat standpoint than from an energy standpoint, from a heat standpoint, breaking bonds is going to require energy. Putting bonds together would release energy. And so if you look at processes that generate a lot of heat, like burning stuff. To burn alcohol, we've got to break some bonds to do that. But we create so many more bonds that happen to be more energetic that the net result is that it feels hot to us because that the recreation of that new stuff generates so much excess energy that we forget about the energy that we had to put in the first place. Now, what's different about ATP and ADP and these hydrolysis processes is that we're not looking at things just from a heat standpoint. We're looking at energy holistically. And holistically speaking, we're going to get energy from this bond breaking because there is going to be some additional energy, excess energy, coming from the fact that we have now separated those two from each other. Again, we won't get into the fine details of it. Just understand that there is a difference between when we look at things from a heat standpoint versus a total energy standpoint. If we were looking at this from a heat standpoint, this would be an energy loser. Breaking that bond from a heat standpoint would require us to input energy. But that's not what we see. We see overall about 7,300 calories for every mole. Now that's little c calories. 7.3 big C calories, food calories. So every mole of ATP that breaks apart gives us 7.3 calories of energy to run processes. Now we can see the same kind of thing happening with ADP converting to AMP. It just doesn't happen nearly as often. And we get the same energy output in total that we did for the first process. So if we think about this is the continuous cycle of metabolism. You start with ADP, you eat something, the energy derived from the digestion of those food molecules allows the ADP to add that phosphate group and turn into ATP. Your body needs energy to function. When it is functioning, the ATP goes, breaks apart in this anabolic process, and we get the ADP back again. And the cycle repeats and repeats and repeats. So long as you are alive, you are going to need this bottom process. Which is why there is such a thing as like base load calorie count. You know, you can't just stop eating because your body needs a certain amount of calories every day just to maintain normal function. Now, if it isn't getting the caloric intake that you need, what starts to happen is it starts to go into a shutdown mode. These anabolic processes start to slow things down considerably. That's where you can hit those kind of energetic lows that come, you know, if you do fasting for long periods of time, or you know, you exhibit other kinds of starvation kinds of behaviors, either intentionally or unintentionally. So let's go through and sort these two molecule sets based upon their functions. So we're looking at ATP versus the ADP and that extra phosphate. Which one of these would be used in an anabolic process? It'd be ATP. Remember, ATP is what goes into the anabolic process that allows the body to perform the function. Which one of these is an energy storage molecule? It's not ADP, it's still ATP. Remember, your body stores ATP in reserve until it is needed for those anabolic processes. ADP is the byproduct of that energy being used. So letter C is basically the same thing as letter A. Anytime we require energy, ATP is what is used. ADP and the free phosphate ion, those are the byproducts of that hydrolysis reaction using up that energy. All right, any questions here out of section 1 before we move on to section 2? All right, so section 2 is all about digestion. In digestion processes, so the first step of metabolism is digestion. And again, digestion is the process that we see where we take those big molecules, your proteins, your carbohydrates, your fats, and break them down into smaller molecules that your body can ultimately use. So what goes on in digestion? Well, couple of things. First of all, the moment you start eating carbohydrates, you actually have enzymes in your saliva that will start to break down some of those glycidic bonds. So remember, the bonds that are being broken down are alpha glycosidic bonds. You look at something like cellulose. So one of the main reasons that we cannot process cellulose is because the bonds there aren't alpha glycosidic, they're beta glycosidic. And our body doesn't have the enzymes necessary to break those down into simpler sugars, which is why they pass through. Right. Yes. So I don't know if I can give an authoritative answer on this. I'll give you a conjecture. Cellulose is one of the primary components of plant cell walls. And so I would imagine that the name cellulose probably has more to do with its origin, its most common location, cell walls, than it does for anything else. Again, I don't know if that's true or not. That's the definition or the explanation, at least, that makes the most sense to me. But I couldn't tell you either. So those alpha glycosidic bonds, the ones that we find in our starch molecules, like the amylose, like the amylopectin, what that breaking down will do is it'll turn those polysaccharides, those starches, into monosaccharides like maltose, like glucose, like dextrins, which aren't monosaccharides, but are definitely smaller than starches are. Once you swallow those carbohydrates and they enter your stomach, the enzymatic action that is taking place stops. And the reason it stops is because the environment in your stomach is now too acidic for those particular enzymes to work. We talked about that in protein, arguing on proteins. Proteins have a very specific window of pHs where they work. Get it too acidic, get it too basic, they stop functioning, or worse, they degrade and denature. So the digestion process for most carbohydrates doesn't really happen inside the stomach. Happens in the salivary glands, or happens in your intestines after the stomach. Because once we get out of the stomach, where the pH was about 2, now we're into the intestines where the pH is somewhere around 8. Now we start to see the production and the use of other enzymes. So for carbohydrates in particular, these are enzymes that we produce in the pancreas. And what they do is they take those remaining dextrins, all of those remaining chains, and they break them down into monosaccharides, into glucose, into maltose. Enzymes on the mucosal walls, that is the lining of the small intestine, the kind of barrier between the small intestine and the rest of the body. That's where we can start to see some hydrolysis taking place, where maltose gets hydrolyzed. We can also see some disaccharides like lactose and sucrose getting broken down into their monosaccharides as well. And it's those monosaccharides that ultimately get absorbed in the intestinal walls, taken into the bloodstream, taken into the liver. And basically anything that isn't glucose, when it gets to the liver, your liver is going to transform into glucose. Glucose is the energy economy of your body. So while you can ingest other kinds of carbohydrates, ultimately what's going to happen inside of your body is that your body is going to transform them into glucose, because glucose is ultimately what is going to drive the metabolic engine. So this is a flow chart I kind of like, just to kind of show what happens. So inside of your mouth, again, we're starting with polysaccharides. Those polysaccharides are usually starches. So again, thinking about amylose, thinking about amylopectin, those starchy molecules that make up the majority of the complex sugars that we intake. Again, cellulose doesn't fit into this description. Cellulose is not something that we can process. It just passes through our bodies as we eat. But those starchy molecules get broken down either into maltose, which is that glucose disaccharide, or into dextrins, both of which are broken down. They pass through your stomach. They get broken down into glucose in your small intestine. If they are glucose based. If they're not glucose based, so like the galactose in lactose or the fructose in sucrose, they pass through your bloodstream, they go to your liver turns the fructose and the galactose into glucose before it enters your cells. So question always comes up, well, why does lactose intolerance exist? It would seem that your body has a pretty good system for taking sugars and breaking them down and allowing you to use them. But for about a quarter of the population, lactose intolerance means that you can't break that down. Why can't you break it down? Well, the primary reason is a lack of the enzyme lactase. And so for people with lactose intolerance, lactase is the primary means by which they either can't do it or they would need to supplement to add lactase to allow them to take in milk and dairy products. And so for people with lactose intolerance, avoiding products with lactose in it is kind of the first and foremost way that they deal with it. Unfortunately, that also means that there are a lot of foods out there that contain milk and milk byproducts that maybe are things that you don't want to avoid. And that's where supplements like Lactaid and other lactase products can come in and provide those people with a temporary boost in lactase to allow them to better digest those foods. But again, it's only temporary because the lactase that is in the supplement passes through the body system as well. It's not a permanent fixture just because you take the lactate. And so that's that's one thing that can be done about that. There have been other kinds of so there have been moves made in refining processes. There has been a move to in some cases create lactose-free milk through the use of putting lactase into the milk. And studies have shown that for those kinds of processing, within 24 hours or so of adding the lactase, somewhere around 90% of the lactose is out of the milk, gone, through the introduction of the enzyme to the milk. Obviously, that's more processing. More processing means more money. That may not be the best option for a lot of people. So that's carbohydrates. For fats, again, with fats, we're talking about triglycerides primarily. Fats are primarily digested in the small intestine. So fats largely pass through your mouth, pass through your stomach. It isn't until the small intestine that they get introduced to anything that can break them down further. And what happens is in the small intestine, this is where they get introduced to bile salts. The bile salts are able to break down the lipid into smaller groups. Those smaller groups are known as micelles. So what happens in your digestive tract, the triglycerides are broken down, hydrolyzed inside of the small intestine. They actually get reformed on your intestinal lining. And this is where we get the use of those lipoproteins that we talked about, where the lipoproteins bind to the so we have proteins that bind to the lipid and allow the lipid to be moved throughout the body. Because unlike carbohydrates, which are, because they are mostly polar based, dissolve well in your water, travel through your bloodstream very easily, lipids are extremely non-polar. And even if we hydrolyze them into smaller pieces, smaller fatty acids, they still are going to be non-polar. And so the function of these proteins, the lipoproteins, is to help with that transport, because the lipoprotein, it would be able to surround the fat and create a globule, which is going to be non-polar on the inside, but polar on the outside. And that exterior polarity allows it to travel into the bloodstream. For proteins, protein digestion actually does begin in the stomach. The pH in the stomach is definitely high enough to denature the proteins, cause them to kind of collapse on themselves, and allow enzymes such as pepsin, which exists in your stomach, to begin to break down those peptide bonds. As those peptide bonds are broken and we get into smaller amino acid chains, your small intestine is able to use other enzymes like trypsin, like chymotrypsin, to hydrolyze those polypeptides and break them down even further down to the amino acid level. And then once they're at the amino acid level, they're actually small enough that they can be transported directly into the bloodstream. as we saw, a good number of the amino acids are polar, whether they are polar and neutral, polar and acidic, or polar and basic. That polarity aids in their ability to move through the bloodstream. So looking at this flow chart here, we can see that the primary means for hydrolysis here, we see a breaking down of proteins into just simply polypeptides in the stomach. The small intestine breaks those polypeptides down to the amino acid level. And once they're at the amino acid level, they can pretty much go wherever they want, wherever they're needed. Okay. Now, this last question is a question that you should be able to answer even if we talk nothing about metabolism today. What are the byproducts of the breakdown of fats, proteins, and carbohydrates? Well, if I look at my options, if I break down a fat, I shouldn't get an amino acid. If I break down a fat, I shouldn't get a glucose. If I break down a fat, I should get fatty acids and glycerol. Kind of the reversing of the process to make that triglyceride in the first place. If I break down a protein, well, what did I use to build the protein in the first place? Amino acids. So if a protein gets broken down, it's going to get broken down back into the thing that made it up in the first place. And ultimately, while not every carbohydrate is made from glucose, we do know that glucose is a simple sugar. So any carbohydrate that is complex, breaking it down to a simple sugar makes the most sense. Now, we just happen to know also that our body works very hard, that the majority of the sugars that we are able to process are glucose-based. So again, amylose, amylopectin, those are glucose molecules that are just strung out and branched out on top of each other, but they're all glucose at their very heart. And the other ones that we can break down, lactose and sucrose, well, half of those disaccharides are glucose as well. The only thing we have to do is break down the galactose or the fructose, and the end of those conversations always end up at glucose as well. So this is where we are going to stop, because again, to go beyond this, we start to get very, very, very deep in the trenches of metabolism and those other processes that we really have no business getting ourselves into as people in this class. So this is where we're going to stop. You're keeping tabs on things. This is also the place where we stop learning new stuff. So